Hello, everyone. I am Panayota from BMKGENE, and I'm thrilled to welcome each of you to our latest webinar. Today, we'll explore integrated approaches in microbiome analysis, ranging from nucleic acid extraction to advanced sequencing technologies. Our webinar today is divided into two sessions. It will last for about 50 minutes, followed by a dedicated 10 minutes of your questions and answers. Feel free to submit your questions through the Q&A button at the bottom of your screen. We're eager to hear from you, and then if any questions are left unanswered, we will contact you to reply to them via email. Additionally, we'll share the slides and a recording of this webinar via email. Now, let me introduce our distinguished first speaker. Dr. Anna Vila-Santa, a field application scientist at BMKGENE with a PhD in bioengineering, brings a wealth of experience in synthetic biology and microbial surveillance. She's well-versed in both wet lab and bioinformatics settings, providing tailored solutions and technical troubleshooting. Today, she will discuss two foundational sequencing methods crucial for microbiome research: amplicon sequencing and shotgun metagenomics. Anna will guide us through a comparative analysis of short-read and long-read sequencing technologies, evaluating their performance for various study objectives. Anna, the stage is yours.
Hello, everyone. Thank you for being here today. It is a pleasure to be able to talk about microbiome analysis and to focus on the process from nucleic acid extraction to sequencing technologies. First, let me briefly introduce BMKGENE. We are a company that provides sequencing services, and we have a multitude of platforms to do this. For short-read sequencing, we can provide Illumina and the NBSEC, while for long-read sequencing, we have available PacBio and Nanopore. We also have the 10x Genomics platform that we use for single-cell solutions. So this means that we are able to cater to multiple research purposes. We can help you in genomics studies, human genetics, metagenomics, transcriptomics, and single-cell omics. I would also like to highlight that we have our own in-house developed special transcriptomics chip called S1000. Finally, we also have a bioinformatics platform available called BMK Cloud. This is on a website and it's very easy to access and user-friendly so that researchers can analyze their sequencing data using their own inputs. Our headquarters is in Beijing, and we have a sequencing center in Qingdao. Since 2022, we also started operations in Germany in our Münster biolab. In our labs, we rely our expertise in nucleic acid extraction, QC, and sequencing using automated systems. This and the fact that we adhere to strict standard operation procedures means that we can guarantee a consistent quality across projects and since we receive the samples to when we deliver the data. I think you'll find during this webinar that having automated systems really is key to obtaining consistent quality. But now this webinar is about microbial communities, so let's dive into that. The first microbial community was reported all the way back in the 17th century. This was in raindrops. Since then, we have been studying microbiomes for a long time. As you can see, I think the most studied ones are probably the human microbiome, the environmental one, and the agricultural microbiome. This is because understanding how these microbial communities work really helps us devise solutions for multiple problems, including health conditions related to mental health, drug metabolism, immune system, but also to environmental and agricultural challenges that we are faced nowadays. This can be key to implementing solutions for pollution remediation or farming practices. So in the past, the way that people have been resolving microbial communities was using a culturomics-based approach. This meant that researchers got the samples and then they would need to isolate the microbes present in them by using different culture media. These culture media were both selective and differential, and so they enabled the identification of the microbes. The problem with this approach is that it's very time-consuming. It takes 24 hours to 48 hours or even more for the microbes to grow and then to passage along the cultures. So this is where sequencing technologies can really help. And they have been applied to microbial studies in mainly three different ways. The first one, called amplicon sequencing or metabarcoding, replies to the question of who are they? So understanding which microbes are present in the sample. Using shotgun metagenomics, we can understand which microbes are present, but also what they can do, what the genes are present there, what is their functionality and possible metabolic potential. And the third strategy, where we can join metatranscriptomics or metabolomics, answers the question of what are the microbes doing in that particular time point. So this means we can find the genes that are being expressed, but also the metabolites that are being produced at that time. In this webinar, I'll focus only on the first two. So let's dive into amplicon sequencing. The idea behind this is to sequence these hypervariable regions of genetic markers. And the most commonly used markers are 16S for bacteria, 18S for eukaryotes, and internal transcriber spacer region for fungi. The most traditional way to do this is to focus on specific regions of these markers. So, for example, in the 16S gene, lots of studies focus on the V3, V4 region. And since this region is under 600 base pairs, this means that we can just sequence them with the short read sequencing, so Illumina and the NBSEC. But in the last few years, the cost for long read sequencing has really gone down. So this means that we can now use PacBio to sequence the full length of these marker genes. And this really improves the outcome of the research projects. To have a closer look at how amplicon sequencing projects occur, I brought here two case studies that were published and that use the BMKGENE sequencing services. In both studies, the idea was to characterize the gut microbiome, so the samples were of mouse gut. In one of the studies, the strategy was to sequence the V3, V4 region of the 16S gene. This was done with Illumina NovaSeq, and for these services, we recommend to obtain 50 to 100K tags per sample, with tags being a number of paired reads that pass the quality filter. In another study, the idea was to sequence the full-length 16S gene. And we do this in-house with the PacBio Revio equipment, and we obtain hi-fi reads. This is important because hi-fi reads have an accuracy that is comparable to Illumina. In the case of PacBio projects, we don't require that much data, so the data output is less. In amplicon sequencing projects, we usually want to understand what is the microbial diversity and how is that different between two samples. So in the first study, the idea was to understand the effect of food supplementation with probiotics in the gut microbiome. And it was found that probiotics result in an increase in the microbial diversity. In the other study, the comparison was between obese mice and mice that are fed with a regular diet. And the conclusion from this data can be that obese mice have a decreased diversity in the microbiome. But besides diversity, it's also important to look at composition. So to understand which species are present in one sample versus the other. So in both studies, this was analyzed, and you can already, I think, see a difference between them, which is in the first study, the taxonomic composition, there are a large percentage of reads that are not classified. While when using full-length sequencing, this is not the case, and you have a smaller percentage of reads that are unclassified. And this leads us to the difference in performance when we do amplicon sequencing with short versus long reads. This can be well illustrated with in silico experiments, such as this one, where a taxonomy tree was constructed using the 16S sequences that are present in the database. And when we are using the entire 16S sequence, which is right here, almost all sequences will be classified. This is because we have the entire information. It's intuitive. But when we start just focusing on this smaller region of the 16S gene, then the percent of unclassified reads really goes up, especially if we are looking at the species level. So basically, with long read sequencing, we can get higher annotation rates at the species level. And we have analyzed this in-house, and this can be a difference of 10 times more annotation, depending on the samples. So let's look at the other technology of shotgun metagenomics. The idea behind this is to extract the DNA content from a sample, fragment it, and then sequence it. Our bioinformatic analysis focuses on taxonomy, so finding the microbial composition, such as what I've shown previously for amplicon sequencing, but now we can also look at function. So we can assemble the reads into contigs, then find the coding sequences in those contigs, and then annotate those sequences with a multitude of databases. And we can use this information to find which functions are more present in a sample versus the other, for example. So some of the databases that we use are focused on the metabolism, so we can find the metabolic networks that are present in the samples. Another example is finding signature or marker genes, such as the determinants of resistance and virulence. And in some cases, in that complex mixture of microbial genomes, we may also want to assemble complete bacterial genomes for that mixture. So again, we have here another case study, again focus on mouse gut samples, and the idea was to extract the DNA, fragment it, and then sequence it in Illumina NovaSeq with paired end reads. The bioinformatic analysis is focused on microbial composition, so this is similar to the 16S amplicon projects, but then again, now we have the function. So eggnog is one of the databases that we use, but we use others such as genontology, KEGG, etc., so that we can find the function, the biological functions that are enriched in one condition versus the other. In this case, the condition was normoxia versus hyperoxia, so exposure to different levels of oxygen. But then again, like it was with amplicon sequencing, with metagenomics, we can also improve our projects by using long reads. So in this other case study that was now focused on the human gut, the strategy included using short reads with Illumina NovaSeq, but then joining them with long reads with Nanopore. And these reads can be 10 to 20 kb. So using this combination, we can do the bioinformatic analysis to research microbial composition and functional composition, like shown in the previous case study. But now we have a plus. We are able to assemble complete microbial genomes. And this is an example from this publication where it was possible to assemble this bacterial genome, but there were others in publication. And we can try to have a look at the bioinformatic analysis that is done to understand why this becomes possible. So when we are doing shotgun metagenomics, we have this fragmentation of DNA, so we have short reads, and then we do a process called de novo assembly, and that enables to create these contigs that usually can be 5 to 20 kb. Then we need to do a process called binning. The idea behind binning is that we have this huge list of contigs, and they are belonging to different species. When we do binning, we basically join the contigs that correspond to a particular species in one group, and then another, and then another. And having these different groups enables us to do the analysis of each genome. The difference when we insert long reads in this pipeline is that now when we do the de novo assembly, we can have a much better assembly. That means that we will have less contigs and they will be longer. So of course, having this longer contigs will improve the binning and will also improve the analysis that we can do afterwards, thus implying that we can obtain complete genomes. These are called metagenomes. So this is all that I had to talk about regarding sequencing. I would just like to take this last minute to focus on our microbial sequencing projects can be designed to yield the best results possible. And for the best results, we need, of course, the best samples, but also the best practices. During this webinar, I focused on sequencing practices, but I would also like to highlight the importance of sampling practices, especially with microbial projects. Host contamination can be a problem. So good sampling really is important. And we provide a sampling guide to our clients to make sure that the samples are the best possible for the projects. And of course, the sample is also really important. So when we're talking about sequencing, we're talking about high quality DNA to have a high purity, a high concentration. And if we are talking about long read sequencing, it's also important that the DNA is not too fragmented. And finally, it's important that the extraction method that is being used will conserve diversity, because if the microbial community is very complex and diverse, we need to make sure that that diversity reaches the DNA stage so that it will appear in our sequencing analysis. And I think Dr. Kui will have a lot to tell us about this too. So thank you for your attention.
Thank you, Anna, for your excellent presentation. We will now be moving to the second session of the day with our second speaker, Dr. Xiao Kui, the product manager of TIANGEN Export Marketing. Dr. Kui is a graduate of the Faculty of Medicine at Lund University, Sweden, specializing in nucleic acid research. Her expertise in human papilloma virus gene regulation and carcinogenesis is backed by published research in top scientific journals. She will provide an in-depth overview of TIANGEN's comprehensive solution for sample preparation and nucleic acid analysis in microbiome research, addressing future challenges and improvements. Dr. Kui, the stage is yours.
Thank you for the introduction. Let me share my screen. Good afternoon, everyone. Thank you for the host, and thanks Biomarker Technology for the invitation. It's my pleasure being here to have this opportunity to learn and communicate with the speakers and audiences. I am Xiao Xu, Product Manager of TIANGEN Biotech Company. Some of you may have used the TIANGEN product before, while some may not be very familiar with us. So before I begin, I would like to give a brief introduction about who we are. Yes, so here is a video.
As life science develops rapidly, researchers need more efficient and convenient tools and methods. TIANGEN has never stopped seeking better solutions to support life science research. TIANGEN has been focused on providing the whole workflow solutions of nucleic acid purification and detection assays, as well as NGS products and customized services for life science researchers in China and around the world. Every day, 95% of Chinese laboratories are using TIANGEN products in over 200,000 experiments. More than 18,000 TIANGEN instruments are serving for scientific research in China. 82,000 published articles cited TIANGEN products up until 2020. We work with scientists to drive research and fuel new discoveries. We cooperate with detection institutes to locate disease threats and detect hidden dangers. We are committed to helping our customers achieve outstanding results and breakthroughs in the fields of research, molecular diagnosis, applied testing, and biopharmaceutical. Our mission is to advance the progress of life science research and improve the application industry chain. TIANGEN has contributed to support fighting against COVID-19 since the beginning of 2020. By the end of 2020, we had provided more than 20 million virus extraction kits, as well as 150 million tests of raw materials for COVID-19 detection. TIANGEN's new production base is located at Zhongguancun Changping Scientific Park, with a construction area of 3,000 square meters. Our whole operations system is under the supervision of ISO 9001 and ISO 13485. All products are manufactured under high standard process. The equipments all come from top global suppliers. A high standard, clean manufacturing environment, stable automated production line, and strict QC processes all act as strong foundations for providing high-quality products. TIANGEN is now recognized as a leading provider of molecular biology reagents, as well as professional technical service to life science customers around the world. We cooperate closely with our partners in multiple ways, including all kinds of customized and OEM, ODM services. Tiangen never stops. We'll contribute to the progress of life science research in China and around the world.
All right. Tiangen portfolio include like 3,000 products in total. We cover sample prep, nucleic acid extraction, and detection product. NGS library prep kit are our pipeline as well, while automated nucleic acid extraction is our focus in the global market at the current stage. Yeah, Tiangen products are widely used in life science research. Over 90% of Chinese universities, research institutes, hospitals, and inspection authorities use our product. So every day, about 200,000 experiments using Tiangen solutions. Okay, then let's come back to our topic today. Growing interest and affordability of microbiome of human and environment approaches for biodiversity assessment and monitoring of complex ecosystem have led to the researching of nucleic acid. Analyzing the microbiome of human and environment has significantly enhanced our understanding on metabolic, physiological, and ecological roles of microorganisms. However, the analysis of the microbiome is affected by experimental conditions, like the extraction condition, the sequencing and data analysis. Moreover, the introduction of new sequencing technology and protocols lead to a flood of new methodologies, which also have an immediate effect on the result of analysis. So today, I would like to have this presentation to discuss some tricky part of microbiome DNA extraction, as well as to provide advanced nucleic acid analysis platform from our side. So here is the outline. We're gonna start from the basic workflow. Genome sequence are usually included in the basic workflow in recent years since it is more informative and help elucidate the function and the role of microorganisms in their environmental community deeply. While the science behind environmental DNA research is complex, the actual workflow of collecting and testing a sample is relatively simple. Samples are collected in an appropriate storage tube or filtered through very fine paper, which traps loose cells and strands of DNA. The techniques to extract the DNA and read what DNA is present are the same as those used for tissue or blood sample, usually like PCR or whole genome sequencing. Scientists can either read all of the DNA present from every organism or target just the DNA from species of interest. Nowadays, even though defined protocol exists, more and more kits are designed especially for microbiome research. There are still lots of difficulties in this field, like the researcher need to consider barriers from DNA recovery to PCR. For example, the humic acid from soil, biosalts from feces, they are all inhibitors, will affect the downstream application. And another feature is bias sometimes makes the result confusing. So why it's so special when we talk about bacteria and fungi DNA extraction, and why this microorganism DNA extraction can be a topic? Because this type of samples, they have cell walls, which makes them more difficult to break open. There are some enzymatic and chemical methods that can get around this, but efficiency will vary depending on species, age, and stage of life cycle. Therefore, to ensure complete lysis, it is best to use a physical method such as bead beating to break down the cells with ceramic beads. Soil sample contains a variety of inorganic, organic, and microbial components. Humic substance in the soil inhibit the PCR process and restriction enzyme digestion, which will affect the subsequent experiment. Nucleic acids are lowly abundant in soil, which makes them more complicated and difficult to extract compared to tissue or cells. Large variation in the appearance, morphology, and conditions of fecal samples. For example, there are like fresh feces, freeze, dry feces, and fecal water. Component variation, quite big. Significant differences in stool composition due to the type of donor, health status, and dietary habits. And there are multiple factors contribute to the wide variation in quality, consistency, and microbiome community composition of different stool samples. So microbiome research typically follows a structured workflow encompassing sample collection, laboratory procedure, sequencing, and bioinformatics analysis. While each location has different environmental characteristics, there are some basic procedure, principles, and techniques for collecting and handling sample that are key for the reliability of a laboratory analysis. In fact, sample collection could be a significant confounding factor that might affect the result and interpretation of a study, including the contamination, transportation, and storage safety. They are all key factors that need to be considered. After we collect the sample, the choice of DNA or RNA isolation method could cause bias during sequencing, which in turn affect downstream analysis. Importantly, the extraction method should effectively capture all types of microbiomes. For example, DNA isolation from gram-positive bacteria is harder due to their thick peptidoglycan cell wall. Traditionally, manual grinding is the most common method used in disrupt a sample, especially for tough tissue. For example, the fungi, yeast, and soil, because tensile strands of the cellular and other polysaccharide compressing the cell wall. Tissue is frozen in liquid nitrogen and then crushed using a mortar and pestle. Another advantage is powder after grinding is suitable for downstream lysis buffer. These two advantages makes it the most classic way to disrupt the thick cell wall. But as long as you have ever tried once before, you will definitely know the weakness of this method. It is time-consuming and labor-consuming. Researchers have to wash, clean, dry, decontamination the mortar and pestle, and our arms are so tired if sample throughput is high. The reproducibility may vary because we need to control the liquid nitrogen volume and grinding time very precisely. So bead-splitting methods are considered to produce superior yield if done optimally. Thus, for a complex bacteria sample, a bead-splitting step could be performed before standard nucleic acid extraction. However, vigorous bead-splitting should be avoided since it can shear nucleic acid and eventually affect the library preparation step later. In order to cover the weakness of liquid nitrogen, TIANGEN have a handle homogenizer which can finish the tissue homogenization by 3 steps within 10 seconds. First, customer load a sterilized plastic pestle by a simple press. Then grinding the sample. Last, trash the used pestle by simple press on the pestle desample box. These accessories makes the whole procedure free from cross-contamination and eliminate the sample wasting significantly. If you have many samples to handle at the same time, here is a homogenizer that can be used to treat various sample types, especially for the microbiomes. Since it can, especially for some RNA and protein extraction to prevent the degradation or denaturation, since it have cooling function to reach as low as -10 degrees. It can process 1 to 24 grinding tubes at the same time with the 3D motion mode to strengthen the homogenization force. It can be very efficient to use with purification kit as downstream. We also provide full range of beads in prefilled tubes to facilitate the use of this instrument. Here, I show a comparison of yeast sample before and after homogenization by this instrument. You can see the grinding is very roughly and even. So after sample pretreatment, I would like to compare several most common purification methods currently used by academic research labs. Phenochloroform is the most traditional method. A mixture of alcohol, phenol, and chloroform is added to the sample lysate to promote the patrition of liquid and the cell debris into an organic phase, leaving the DNA in the aqua phase. As an alternative to the phenochloroform procedure, the spin column method has the advantage of not using toxic chemical. Nowadays, most commercial reagent kits use spin column method, which is a simple bind, wash, elute process. Nucleic acid bind to the silicon membrane in the presence of high salt. Polysaccharide and protein do not bind well to the column, and residue traces are removed during alcohol-based wash step, along with the salt. This method requires multiple centrifugation or vacuum step to flow liquid through from the membrane. And so recently, magnetic beads-based methods are more and more adopted by researchers. Because this method also employed an easy bind wash elute process, the DNA and RNA binds to the paramagnetic beads added to the sample instead of the silicon column. Magnetic beads are conjugated with ligands that specifically bind DNA, RNA, like the nucleic acid. Using a strong magnet, the nucleic acid-bound beads are transferred into the wash buffer to remove unwanted material. After washing, the genomic DNA or RNA is eluted from the beads in water or a local buffer. The beads-based method is scalable and automation compatible. So researchers usually select the method that best suits their needs based on the advantage and disadvantage among the three methods. After we select an appropriate method and make an excellent extraction and purification, the next step is usually quality control. The golden method to evaluate the quality of nucleic acid before is using electrophoresis. TIANGEN offers a device to illuminate and visualize the nucleic acid or the protein, facilitate gel cutting. The conventional transilluminator uses the UV light. Exposure to UV can be dangerous to human skin as well as the samples. To avoid those issues, we instead use blue light, that is safer for human and the sample. Furthermore, nanodrop and qubit are frequently used to measure the concentration and purity since it is very convenient to the customer. For sequencing part, ring value are more informative since it can give us the integrity and precise concentration and purity. Some researchers even use qPCR for validation before the final sequencing. So as of my last knowledge, the field of microbiome analysis was rapidly evolving, and the new challenges and advancement may have occurred since then. As of that time, here are some challenges associated with microbiome analysis from the experimental part to computational part. So with 20 years of experiences in nucleic acid extraction, we have found the most suitable magnetic beads and buffer system for most samples used in scientific research. We modify the physical and chemical properties of the beads to suit different purification needs, from total RNA, genomic DNA, to PCR or gel purification. When used with our optimized buffer system, it can produce high yield, purity, and the integrity from the majority of scientific research sample types. We have prepacked reagent and prefill cartridge to use on automated nucleic acid extractor for maximum efficiency and minimum hands-on time. So this small instrument, the TIGAT S16, is groundbreaking due to its unprecedentedly compact size without compromising quality. It's time-saving since it minimizes researchers' hands-on time, thereby increasing the efficiency and providing comfortable experiences. It is an out-of-the-box automation, little installation and training required. There are pre-installed programs you can select from, no external laptop, software, or barcode scanner required. Everything can be operated on the building's touchscreen. We provide specialized solutions for most of the sample types. So working with TIGAT S16, it only takes 3 steps to purify nucleic acid. The researcher adds the sample, slice it into the pre-filled reagent cartridge, choose the program, and the last one is start the run. In the meantime, you can work on other tasks or have your lunch. After 30 minutes to 1 hour, depending on different kit, the purified nucleic acid are eluted in the 5th column. You can transfer the DNA or RNA into a new epidermal tube for long time storage. So here you see some figures. There are a screen here, and customer need to prepare the sample and our cartridge. Then load the sample to the cartridge. The last step is to choose a corresponding pre-installed program and then press start. And then this small instrument will take over the rest of the purification work. So based on our marketing feedback, the animal tissue cell DNA RNA kit, especially soil stool microbiome DNA kit, are the most popular ones. We are keep collecting data and sample types from our customer side. And we are in developing the environmental kit. This kit is upcoming very soon. So this slide is some data obtained from the environmental sample, the kits that I mentioned before, such as animal feces, soil. The most obvious advantage of this kit is the purity. The optimized buffer system largely help get rid of the inhibitors from sample, which lead to the downstream application, like PCR, more easily. And here is the 16S sequencing result compared to the Zymo standard. The closer the percentage is to the Zymo standard indicates a smaller bias in extraction. A small bias indicates that this product has no preference and is homogeneous in its ability to extract the various bacteria in environmental samples. Except the soil and stool sample, the environmental samples are even more complicated. We are keep trying to cover as much as we can, including some very, very tricky sample types, like the garden soil, lake mud, animal mature ferment broth, dust swab, soil salts, wastewater, and some intestinal content from fish or other types of species, mold culture, tobacco material. We are collecting data. So the reproducibility and unbiased of TIANGEN products is as good as the top global brand. For some sample types, even better. We try to provide a comprehensive solution system that can cover as many sample types as possible. Currently, we provide the reagent we listed here. It covers soil, cell, tissue, blood, plant, soil, fecal samples, gel, and PCR product purification, cell-free fluid, bacteria, urine, oral swab, and dry blood spot. And we also plan to launch plasmid extraction kit, FFPE extraction kit, and bacteria, fungi, RNA extraction kit. So TIANGEN has been selling automated nucleic acid extractor for more than 10 years. From 2012 to 2019, the target segment of our automated nucleic acid extractor is the clinical diagnostics market in China mainland. And those extractors feature high throughput and high performance cost ratio. In 2021, to cater the needs of overseas scientific research market, we designed S16, which is compact in size, small to medium throughput, and is compatible with a wide variety of sample type. The S16 is combination with our supporting reagent kit, which can meet 90% of nucleic acid extraction need in lab. Besides S16, we also plan to launch S32 Pro and S96 Desk in the next future. They have higher throughput to meet the needs of our industrial customers. So here I list a few benefits of automated extraction. The most important benefit is the consistency and reproducibility of the isolated nucleic acid. Manipulation of the sample and reagent is reduced, which dramatically decreases the chance of cross-contamination. And second one is safe and environmental friendly. There's no need to use toxic reagent or organic solvents such as the chloroform. And the last one is high throughput automation, save labor and time. Okay, thanks for your listening. This are all the messages that I want to introduce to all of the customers.
Thank you very much, Dr. Cui, for your very informative presentation. And we will now be moving to the Q&A session of today. We have already received some questions, but feel free to press the Q&A button and submit more questions. Okay. Starting with the first that I can see, I don't have much experience with microbiome sample pretreatment. Do you have a standard program for different kinds of samples? I think that's addressed to Dr. Cui.
Okay, yes, I can make some comments of this question, because I think this really very important and tricky part for the sample pretreatment part, because sometimes the samples are quite difficult to break down, and the customer need to use a lot of time to optimize the pretreatment method. So since we have a lot of experiences about the homogenizer, we have already collecting more than 100 program, the standard program that pre-install in the homogenizer. So we can recommend our customer a very precise procedure, like the bead splitting time is 30 seconds, and the rest of the time is 30 seconds. And you have to make this cycle like 3 cycle or 6 cycle. And the strength is from 1 to 6. So we have already collected around 100 preloaded program. So we can recommend our customer a specific program according to a specific sample type. That will save a lot of optimization time for our customer.
Thank you very much. The next question is for Anna. What is the difference in cost between amplicon with Illumina and PacBio?
Of course, the difference may vary between projects, but I can give a rough estimate that I think it can be less, can be around like €3 per sample doing PacBio full length instead of short reads. So I think it's really worth it from the advantages that you get out of it.
And there is another, this one is for metagenomics. In metagenomic studies with Nanopore, is it really important to also perform Illumina sequencing?
Yeah, so I didn't address this so much during the presentation, but the idea is, so Nanopore is great because it provides long reads, but it has a problem with accuracy. It's much lower than Illumina and PacBio. So this means that when we do metagenomics with Illumina plus Nanopore, we can use the Nanopore long reads to do the assembly, but we need the short reads from Illumina to do error correction. That's why we need to use both at the same time.
Thank you. And let's move to the next one. This is for Dr. Cui. I was wondering about cross-contamination, considering this instrument is an open system. How can it be prevented?
Yes, so we have tested cross-contamination since the throughput is 16, and we do like 6 rounds of the experiment to test if there is cross-contamination. The result is, even though it is a open system, but we have a pre-installed fan which can evaporate the air from the cabin to outside the door. And the strength of pipetting are optimized to a precise precise position, which can prevent the splash of the liquid. So our testing result is, even though this is a open system, it still can prevent cross-contamination. And another advantage is this small instrument have a pre-installed UV light. If customer want to do like the denaturation of the nucleic acid, the customer can choose 30 minutes to 40 minutes by using this UV light. Yes.
Thank you very much. We have some more questions, but in order to save some time, we are going to conclude today's session right now. I just want to express our gratitude for your participation. We are going to answer all your questions via email. Look out for this email because it will also have the recording and the webinar summary. And also check out our social media pages for both companies so you can follow our contact and learn about future webinars. Thank you very much again and have a nice day.